Hello everyone, welcome to masterspharma Academy. Hope you are doing great. In today's session, we will discuss about some pharmaceutics MCQs. The first question. The process of reducing particle size using mortar and pestle is called trituration, ligation, pulverization by intervention and comminution. The correct option is trituration. Trituration refers to dry grinding by solid substances in mortar and pestle. The main purpose is to reduce particle size, thus when only mortar and pestle are used. In question, they specifically mention mortar and pestle, so the correct answer is trituration. The second question, zeta potential indicates flocculation tendency, particle size, viscosity and osmolarity. The correct option is flocculation tendency. Here, what is zeta potential? Zeta potential is the electrical potential at the shear plane of dispersed particle in a colloidal system. It reflects the surface charge, therefore, the stability of suspension or emulsion. Why zeta potential indicates flocculation tendency? Particles in a suspension repel each other due to surface charges. Higher zeta potential is equals to stronger repulsions and more stable system and less flocculation, whereas lower zeta potential is equals to weak repulsions, particles come close and flocculation occurs. The third question, bioavailability is the highest for oral tablets, capsules, IV injections and suspensions. The correct option is IV injections. What is bioavailability? Bioavailability is the fraction of administered dose that reaches systematic circulation unchanged. Mathematically, F is equals to amount reaching systemic circulation by amount administered. Why IV injections has the highest bioavailability? IV route drug directly into the blood stream. There is no absorption, no dissolution, no membrane transport and no first pass metabolism and no degradation. IV administration is equals to 100% bioavailability. All the other routes have less 100% due to absorption barriers and first pass metabolism. The next question, direct compression requires large dose drugs, high flow and compressible excipients, surfactants and wet massing. The correct option is high flow and compressible excipients. What is direct compression? It is a tablet manufacturing method in which powders are compressed directly into a tablet without any granulation steps. For this method to work, the powders blend must have the first one, excellent flow properties. So it can uniformly fill the die cavity and prevent weight variation. And the second one is high compressibility. So it can form a strong, stable compact when pressure is applied. So therefore, only excipients with superior flowability and compressibility are suitable for direct compression. For example, spray dried lactose and microcrystalline cellulose. The next question. The most commonly used capsule materials is chitosan, gelatin, PVA, polyvinyl alcohol and the PVP, polyvinyl pyridine. The correct option is gelatin. Gelatin is by far the most widely used material for manufacturing hard and soft capsule in pharmaceutical industry. 90 to 95% of all capsule shells are used globally. Why gelatin is the most common capsule material? Excellent film coating properties, safety, biocompatibility and non-toxic, rapid dissolution at body temperature, ease of manufacturing and compatible with most drugs. The next question, zero order drug release means concentration dependent release, constant release rate, no release, delayed release. The correct option is the constant release rate. Zero drug order release is an ideal type of drug release in controlled release formulation. In zero order kinetics, the drug released at constant rate per unit, completely independent of concentration. The key principle of zero order release, rate of drug release is equals to constant is equals to K. This means the same amount of drug released every hour Plasma drug levels remain stable and avoid toxicity and subtherapeutic level. The next question. Lyophilized products show low stability, long reconstitution time, excellent stability and no sterility. The correct option is excellent stability. Lyophilization is a freeze drying, a specialized drying technique used for sensitive biological and pharmaceutical products. It removes water by sublimation under vacuum, leaving behind the porous and dry solid. This process used specifically to increase the stability of drugs that degrade in aqueous solution. While lyophilized products have excellent stability, removal of water and it prevents of hydrolysis, low temperature drying and no thermal degradation, and it has a long shelf life. The next question. The term uniformity of content applies to tablets, capsules, low dose potent drugs and high dose drugs. The correct option is low dose potent drugs. Uniformity of content is a critical quality control test required by pharmacopoeias for low dose potent drug products. The test ensures that individual dose unit contains the correct amount of active drug within a narrow range. Why it is applied specifically at low dose potent drugs? When a drug is present in a very small amount, typically less than 25 mg or 25% of total tablet weight, even a minor mixing of manufacturing variant causes high dose variabilities, therapeutic failures and toxicity because active pharmaceutical ingredient amount is so small, weight uniformity alone cannot guarantee correct drug content. Uniformity of content is mandatory for potent drug, low dose drugs and narrow therapeutic index. The next question. Phase separation in emulsions is called caking, cracking, creaming and the sedimentation. The correct option is cracking. Emulsion instability can appear in several forms, but phase separation, where the oil and water layers completely separate, it's specifically called cracking. Why the cracking is correct? Cracking irreversible phase separations. The dispersed droplets lose their protective films and cannot be stored by shaking. The next question. Controlled release system reduces dose frequency, bioavailability, off-life and patient compliances. The correct option is dose frequency. Controlled release or modified release drug delivery systems are designed to release the drug at predetermined substantive rate over an extended period because the drug is maintained with the therapeutic window for a longer. Dose frequency significantly reduces. For example, a drug that normally requires dosing 3 to 4 times a day in immediate release form may be given once daily in controlled release form. This is for the today's session. Thank you for watching. For more such content, please consider subscribing and follow us on Instagram masterspharma. Thank you.